Okay, so for this one, we'll be talking about proteins. Specifically the primary structure of the proteins. As you know, proteins are made up of amino acids, and the basic structure of the amino acids are, first, you have a central alpha carbon, which is linked to a carboxylic acid group. Which is linked to an R group, a hydrogen, and an amino group. And an amino group. Okay, so basically proteins are made up of peptide bonds. So a peptide bond is when two of these are joined together. So imagine another amino acid joining to form a peptide bond. So the mechanism for this is, okay, remember back to organic chemistry too, when you have a carboxylic acid, and imagine you're adding it to an amino acid, this is NH3. So basically what it is since oxygen is an electronegative atom, there's a dipole moment, so all the electrons are going towards the oxygen. So there's a partial negative and a partial positive sign on here. So if you have a negative, if you basically have a p orbital that has electrons, so the electrons will get attracted to this positive sign by Coulomb's law. And then when that happens, all the electrons get shifted onto the oxygen at first, and they get downshifted, and this oxygen, or this hydroxide group leaves. So what you have here is H3. But there will be a positive sign because you added something, and in order to get rid of that positive sign, this hydroxyl group that you just released will come in. So since the hydroxyl group will be negative, and another way that you can test this out was because you have a negative plus a positive, that makes up 0, and if you look on this side, there's 0 charge. So that's another way to make sure you're doing it right. And so this hydroxyl group comes in and it will take one of the hydrogens out, so you will just be left with this structure right here, NH2. And that is basically an amide. So this is basically what really happens, and just to make it much more clear, I will just do another in terms of an amino acid. So suppose you have this right here, the carboxylic group, you have the alpha carbon, the hydrogen, the R group, and this will be N, H, and we'll just say this is a H. Usually I'll go later into this, this actually loses an H, and this has an extra H at the end. When it's in actual water, you have ionic, it's actually in an ionic state, but we can discuss that later. For the essence, this is just to make sure that you understand the amide group formation. So this is one amino acid, and then this will be the other amino acid. The R group and the amino acid. Just some terminology. This is called the C terminus because this is the free carboxylic group, and this is called the N terminus. So this is the C terminus and this is the N terminus. Okay, so what really happens is the same thing that we went over here. You have the nitrogen electrons, and the p orbitals that contains the electrons in this nitrogen atom comes in and attracts the partial positive oxygen, or partial positive carbon. So basically this comes and attacks this, and the same thing happens. All the electrons get transferred here, then they get transferred back to reform the double bond, which causes this hydroxyl group to leave. And then this hydroxyl group comes and it takes out one of the hydrogens. So in essence, you have lost one hydroxyl group, and you have lost one hydrogen. So that just makes up water. And this is why we call this a condensation or a dehydration reaction, because you're losing water to form a peptide bond or an amide bond. So you have the end product will just look like this. Remember, since you lost a hydrogen, you will only have one hydrogen. You have this bond, carbon, since you lost this hydroxide, so you lose this and you lose this. You have the initial carbon, alpha carbon, the hydrogen, the R group, and the amino intact, right? So just to make sure, this right here is what's called a peptide bond. In a sense, this is this arrow right here, right? And this group, this is called an amide group. So this is basically joining two amino acids together, and when you have a string of amino acids, that's called its primary structure. And yeah.